So, the Oxford iGEM team will be the next team to present their talk about DCMation. Have fun.
So, good morning, ladies and gentlemen. I'm Sean. I'm a lawyer on the Oxford iGEM team. And together with Glenn, our biochemist, and Oliver, our engineer, I'm proud to present to you this morning on behalf of the first ever Oxford iGEM team, DCMation, which is a revolutionary biodisposal mechanism for chlorinated solvents, using dichloromethane, or DCM for short, as a case study. Our team's been entirely student-led, from the ideas brainstorming stage to the Jamboree today. And our broad range of disciplines has enabled us to explore all different aspects of our project. And we were excited by the possibility of using bioremediation and a synthetic biology solution to environmental problems. And to this end, we approached the UK Environment Agency, who highlighted the problem of chlorinated solvent pollution, to which there's currently no satisfactory, environmentally sound disposal mechanism. This means that clean-up operations in the UK alone cost millions of pounds every year, and in other parts of the world are not so fortunate to be able to afford to regulate contamination and clean up pollution. And we hope that DCMation could offer international benefits by providing a cheap and accessible solution to the problem of chlorinated solvents in these areas of the world. You may not know it, but for sure you've come across chlorinated solvents in your day-to-day life. They're essential for a broad range of industrial, professional, and even home use applications in products such as paint stripper, spray paint, fire extinguishers to pesticides, and are also widely used in chemistry and other laboratories. Unfortunately, despite the fact that chlorinated solvents are a toxic pollutant, harmful to health and the environment, and a known carcinogen, disposal is frequently careless. It contaminates water systems, and with a half-life of over 700 years in water, this is a massive environmental problem, causing asphyxiation and cancer in biological organisms. There's also a link with photochemical smog and acid rain when DCM is broken down in the atmosphere, and a possible, though underexplored, link with global warming, which is pretty shocking stuff. Our work with the environment agencies demonstrated a clear need for a SynBio solution to this real-world problem. But for our project to have this impact beyond the bench, we needed to constantly consider the practical implementation and safety aspects of our project, as well as ensuring constant communication both with the general public and between the different disciplines within our team. And these considerations influenced every aspect of the wet and dry lab development of our project. As I'm going to hand you over to Glenn and Oliver to explain.
So with that, I give you DCMation. And we envisage this all-in-one disposal kit to work as such, with DCM added into the top and sits at the bottom of an aqueous layer, where it can partition into it. There, it's broken down by our DCM degrading bacteria, which is contained into our novel approach to containment in our biobeads. And we'll also work towards building a DCM biosensor that will be able to report on the presence of DCM and thereby indicate its complete degradation and therefore can be disposed of. To move towards this, we split our project into 3 strands, the first of which to choose and test our chassis, next to construct a biosensor, and finally to optimize DCM turnover in our chassis. Initially, we took forward several bacterial strains, including many Pseudomonads, and found that ultimately E. coli could survive up to 100 millimolar DCM with no inhibition of growth. So that was more than plenty for our purposes. To move towards creating a biosensor, we needed to look at where our parts could come from. And we wanted to find a bacteria that could grow on DCM, and we could probably assume that in there we'd find some sort of DCM-modulated regulatory network. Indeed, when we looked at Methylobacterium exctorquens, strain DM4 here, we found exactly that. This native gene cluster exists, where we have DCM-R in one direction and DCM-A in the other, both driven by promoters present in this intergenic region. DCM-R is a regulatory protein and is thought to be modulated by DCM, which is perfect for us. DCM-A, on the other hand, is an enzyme, a dichloromethane dehalogenase of the glutathione S-transferase family, converting 1 mole of DCM into 1 mole of formaldehyde and 2 moles of hydrochloric acid through 2 toxic glutathione intermediates, shown as X and Y here. So if we're wanting to make a biosensor that's sensitive to DCM, it seems obvious that DCM-R is what we should be looking at. So we set about building this construct here, which involves driving expression of TETR under a constitutive PM promoter. A TETR is a repressor protein that will then bind to this yellow PTET promoter and inhibit and repress expression of any downstream genes. The addition of ATC, or anhydrous tetracycline, relieves this repression and allows our downstream genes to be expressed. In our case, since we're interested in detecting DCM, the gene of interest for us is DCMR here. But indeed, a researcher could insert any gene they like here and have full ATC-inducible expression totally independent of the cell type they used, or any chassis. So we wanted to model how this expression of DCMR would work under various ATC concentrations. To facilitate this model, we made a translational fusion to mCherry, and Ollie will explain this now.
Okay, so to model the ATC induction of the DCMR gene, we use this equation here. This states that the concentration of DCMR protein present in our system is given by an activation due to ATC, the degradation of the protein itself, and a basal transcription rate due to promoter leakage. We were able to use this steady-state DCMR mCherry expression data to fully parameterize our model, also using the method of least squares. This then allowed us to calculate every one of these parameters and gave us the exciting possibility of being able to predict the future, with allowing us to predict the dynamic response of the system. So our deterministic prediction is shown in red here. Our one realization of our stochastic prediction is shown in the blue line here. And we were happy to find out that when we were able to run the experimental data, get the experimental data for this graph, it fitted quite closely as shown. The next step would obviously be to obtain lots of these time courses and to tweak our parameters and make the model predictions even closer.
Okay, so we now know how DCMR is going to be expressed when we add certain amounts of ATC, but where does DCMR bind? So we go back to Methylobacterium extorquens here, and it's this intergenic region that the literature suggests is where DCMR binds. Taking this, we constructed these two constructs you see here, isolating expression from pDCMR in one direction, and then another to isolate expression from pDCMA. Putting these two constructs that we've spoken about together, we can see here that this is the synthetic circuit we've made, and it's at the heart of what our biosensor will be. So we have DCMR fused to mCherry under anhydrous tetracycline control, which then interacts with either the pDCMA or pDCMR promoters, driving expression of our output signal, GFP. This regulatory network between DCMR, that regulatory protein, the solvent DCM, and the two promoters is currently unknown, and so the first step in moving towards our biosensor is to characterize this.
We suppose two hypotheses. The first is that DCMR acts as an activator on both the pDCMA and the pDCMR promoters, and the second hypothesis is that DCMR acts as a repressor on the pDCMA and the pDCMR promoters. Using similar equations to the one I explained earlier, we are able to use a modeling prediction of what we can expect for both these hypotheses. If hypothesis 1 is correct, we expect to see an increase in the fluorescence when in the presence of DCMR. And if hypothesis 2 is correct, we expect to see a large decrease in the GFP fluorescence in the presence of DCMR. This should hopefully allow us to use the presence of DCMR to test between these two hypotheses.
So using this, and just to recap the two constructs that we've built, we ran some wet lab experiments and saw that indeed in both scenarios with both promoters there is a significant decrease in expression upon addition of the regulatory protein or expression of the regulatory protein DCMR. So therefore the conclusion is that hypothesis 2 is correct and DCMR is a repressor at both of these promoters. So now we know how DCMR works, the next question then becomes, how does the solvent DCM interact with this? So we isolated both promoters again and ran some experiments looking at the expression of GFP both before and after DCM was added. Looking at the pDCMR promoter, you can see that there seems to just be no change in fluorescence with and without the regulatory protein present. So we can assume this genetic circuit exists, where DCM has no influence at all. But interestingly, when we look at the pDCMA promoter, we do see some changes. And with the regulatory protein present, we do see an increase in expression of GFP, which fits in well with what we might expect